Hello, everyone, and welcome to our screencast on nutrition, metabolism, and body temperature regulation. One of the problems that we run into every time we try to talk about nutrition is that we basically have 1 week or about an hour and a half worth of class time to talk about nutrition. That presents a problem. I also, from time to time, for Mount Aloysius College, teach nutrition, and whenever I do, it's a 15-week course. So how do we take what potentially is a 15-week course and kind of boil it down to something we can talk about in an hour and a half? Well, that's kind of difficult to do. And that certainly tells us that we can't cover everything there is to cover in nutrition. So for this class, for healthcare professionals, what I've tried to do is to set up this PowerPoint to take a look at nutrition from the standpoint of diabetes. And so every one of you is going to wind up encountering patients. If you get out there into the workforce, you're going to encounter patients who are going to be dealing with diabetes. Why? Well, let's take a look at some facts regarding diabetes. Nearly 30 million Americans currently have diabetes. And diabetes and prediabetes cost the American healthcare system about $322 billion per year. 86 million Americans have prediabetes. One in five dollars of health care is spent caring for people with diabetes. And today, 3,835 Americans will be diagnosed with diabetes. Today, diabetes will cause 200 Americans to undergo an amputation, 136 to enter end-stage kidney disease treatment, and 1,795 to develop severe retinopathy that can lead to vision loss and blindness. You are certainly going to deal with many of these patients as you enter the profession. So it's important that we take a look at nutrition from this particular standpoint. So we know that diabetes is a condition in which we have elevated blood sugar. So let's take a look at how, whenever we eat food in our diet, how sugar is processed in the blood. So the sugar is going to enter the blood, and then it has to be passed from the blood into the various cells of the body. To enter the cells of the body, you know that glucose can't be broken down into smaller pieces, and that the glucose doesn't pass easily through the plasma membrane of cells. Therefore, glucose is going to need some help to get across the cells. And that takes us to the concept of facilitated diffusion. So you may recall facilitated diffusion from A&P1. And if you don't, we're going to take a look at a video of facilitated diffusion in just a minute. But facilitated diffusion is the method by which the glucose gets across the plasma membrane of cells and can enter cells so that they can use it to carry out cellular respiration and thus produce the energy they need to carry out their chemical reactions. So let's now take a look at a video on how facilitated diffusion, thinking specifically of glucose, how facilitated diffusion occurs.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor, which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, The same carrier protein would then transport the molecules out of the cell.
Okay, so now that we've looked back at facilitated diffusion, when it comes to glucose, there actually is what we call a wide spectrum or a wide variety of different types of glucose transporters. I think 12 to 15 glucose transporters have already been identified, and it's suspected that there are even more. Now, we don't have time in this class to be able to talk about all the different glucose transporters there are, but for our discussions here, there are 4 that are important, and we're going to take a look at them. The first glucose transporter we're going to look at is called GLUT1. GLUT1 is a glucose transporter that's responsible for the basal glucose uptake required to sustain respiration in cells. So all cells have GLUT1 transporters embedded in their plasma membrane, so that when glucose is present in the blood, it can be transported into the cells and they can use it for their basic energy needs. In addition to GLUT1, there's GLUT2. GLUT2 is a bidirectional transporter. It is found in liver cells and the beta cells of the pancreas. It's found in the epithelial cells in the mucosa where absorption is important in the small intestine. And it's also found in renal tubular cells. That is to say, it's found in certain cells that are important in urine formation in the kidneys. These bidirectional transporters allow the glucose not only to enter the cell through the plasma membrane, but will also transport glucose back out of the cell and into the interstitial fluid and eventually into the blood. A third glut transporter, glucose transporter, called GLUT3, is one that's expressed mainly in neurons. It has a high affinity for glucose, which allows cells to transport glucose even at times of low concentrations. So even at times when glucose levels are low in the blood, these GLUT3 transporters will allow neurons to transport glucose in so that the nerve cells and the brain cells particularly can continue to be active and protect those cells and give them the energy that they need. The fourth one, GLUT transporter we'll take a look at here is GLUT4. GLUT4, and this is the one that's going to be important in the rest of our discussion, is a glucose transporter that's found in adipose tissue, skeletal muscle tissue, and cardiac muscle tissue. It is what we call the insulin-regulated glucose transporter. This one is tied to insulin production, and insulin controls this transporter and its ability to get sugar in and to get sugar out of the blood and into these various tissues, adipose, skeletal, and cardiac muscle tissue. Okay, with that in mind, then, we need to take a look at insulin itself, because insulin is very important it's a hormone, and it's very important in cells' ability, particularly adipose, skeletal, and cardiac muscle cells' ability to absorb glucose from the blood, therefore having a great effect on blood sugar levels. So let's take a look at the physiologic effects of insulin. Insulin, we say, is the key player in the control of intermediary metabolism. Now, what's intermediary metabolism? Well, intermediary metabolism is the chemical reactions. Remember, metabolism is the sum total of all chemical reactions that occur in your body. The intermediary metabolism is the chemical reactions that actually occur in cells, particularly those chemical reactions that are related to energy production and use. So insulin has profound effects on both carbohydrate and lipid metabolism, as we'll see, and it will also significantly influence protein metabolism and also to that end mineral metabolism. So as we continue on, we're going to break it down for you, and we're going to take a look at the effect of insulin on carbohydrate metabolism first. Then we'll look at lipid metabolism and the effect that insulin has on it. and we'll spend the remaining time working on protein metabolism. We're not going to spend a whole lot of time on mineral metabolism here in this PowerPoint. Okay, so if insulin is very important, insulin is a hormone. It's produced by the pancreas, and in order for hormones to have their effect, they have to attach to receptors on or inside of cells, you may recall from the endocrine system discussion. So let's take a look at the insulin receptor. The insulin receptor is a transmembrane or integral protein. That is to say, it's one that, as we see in our diagram over here on the right-hand side, integral proteins are ones that pass entirely through the lipid bilayer of the plasma membrane of the cell. This transmembrane or integral protein is activated by the presence of insulin. The insulin receptor actually consists of two subunits, referred to as an alpha subunit, as we can see here in yellow, and the beta subunit, as represented in blue. The alpha subunit contains the insulin binding site. So insulin would be able to bind right here and here to the alpha subunits. And when it does, it's going to exact changes that are going to occur inside the cell. We'll be taking a look at what those changes are in just a bit. The beta subunit, however, penetrates through the plasma membrane and really just kind of acts as an anchor to hold the entire unit into the plasma membrane. So once insulin then binds to the insulin receptor, this is going to stimulate changes that are going to occur inside the cell that are going to cause the cell to take up glucose. All right, let's break it down. Let's begin with how insulin affects carbohydrate metabolism. You may recall that carbohydrates are made up of carbon, hydrogen, and oxygen only. And so that's why an appropriate abbreviation for carbohydrate is CHO. You'll see me using that throughout the PowerPoint. Okay, so glucose is liberated, we know, by the digestion of dietary carbs in the small intestine. Amylase and other enzymes break down carbohydrates into their building block units. The building block units are glucose. The glucose is then going to be absorbed into the blood. This is going to increase blood glucose levels. This increase in blood glucose levels is going to stimulate certain cells in the pancreas called the beta cells, and they're located in the pancreatic islets. And these beta cells will be stimulated to produce and secrete insulin. So an increase in blood glucose levels above average is about 90 milligrams percent. When it goes much above 90 milligrams percent, these beta cells then record that the blood glucose level is too high, and they begin to produce insulin. They secrete the insulin into the interstitial fluid. It enters the blood and now begins to circulate through the blood. So the insulin acts on cells throughout the body as it passes through the blood. Any cells that have insulin receptors on their surface will be stimulated when insulin binds to those receptors to take up, utilize, and or store glucose from the blood. The result, we say, is going to be a hypoglycemic effect. In other words, the blood glucose level is going to come back down again. So when we eat these dietary carbs, the carbs are broken down into glucose. The glucose is absorbed into the blood. As the glucose is absorbed into the blood glucose levels start to go up rapidly. Beta cells record this rapid increase in the glucose. They begin to secrete the insulin. The insulin enters the blood, binds to insulin receptors, which are usually found on adipose, skeletal muscle, and cardiac muscle cells, and some other cells. And the net result is that the blood glucose level is going to come back down again. That's a hypoglycemic effect. Okay, so Insulin then, we say, facilitates the entry of glucose into muscle, adipose, and several other tissues, which would include liver cells as well. GLUT4, remember the 4 GLUT transporters we just talked about, GLUT4 is the major transporter for the uptake of glucose in these cells. So all cells will have, let's say, for example, GLUT1 transporters permanently embedded in their plasma membrane. But certain cells, like muscle and adipose cells, have additional GLUT4 transporters. These GLUT4 transporters, however, are not always present in the plasma membrane of the cell. In the presence of insulin, and once insulin binds to the insulin receptors on these muscle and adipose cells, it stimulates changes in the cytoplasm that causes the GLUT4 transporters, which are held in vesicles inside the cytoplasm, to be transported to and inserted in the plasma membrane. This adds additional gates that can allow glucose to enter these cells at a more rapid pace to help bring the glucose level back down again. In the absence of insulin, GLUT4 glucose transporters are present then in cytoplasmic vessels where they're useless for transporting glucose. When insulin binds to the insulin receptor, this is going to lead to a rapid fusion of GLUT4 vesicles with the plasma membrane. They're going to be raised up and inserted into the plasma membrane, as I said. This is going to help bring the blood glucose levels back down again. When the blood glucose levels of insulin decrease and the insulin receptors are no longer occupied, the glucose transporters will be pulled back out of the plasma membrane and recycled back into the cytoplasm, where they'll be held in vesicles until insulin levels go back up again. Let's take a look at a video on how this works.
Diabetes type 2 is the most common form of diabetes. Although it can occur at any age, it is more common in people who are overweight, sedentary, and over middle age. Glucose is the main source of energy for body cells. When blood sugar levels rise, as is normal after a meal, the pancreas gland, located behind the stomach, secretes the hormone insulin. Insulin enters and circulates in the blood and acts on the insulin receptors present in muscle, fat cells, and other tissues of the body. Binding of insulin to these receptors causes the glucose transporters to come to the cell surface. This facilitates the entry of glucose into these cells. In diabetes type 2, the production of insulin is low, and sometimes there may be resistance to insulin. The circulating insulin fails to facilitate the absorption of glucose into the cells and to keep the blood glucose level at optimum levels. This results in the rise of blood glucose levels. The excess in blood glucose reacts with proteins in tissues to form what are known as advanced glycation end products, or AGEs. AGEs create an inflammatory condition in the vasculature, which causes heart disease and damage to other organs, such as the kidneys. Diabetes can eventually cause damage to the heart, arteries, kidneys, nerves, eyes, and skin.
Okay, so you can see the importance of these GLUT4 transporters and why I've chosen to talk about diabetes in our nutrition discussion here. Let's continue on now. So let's take a look at the effect of insulin on glucose metabolism. So insulin, also there are insulin receptors on the surface of liver cells. And when insulin binds to those receptors, it's going to stimulate the liver cells, to take up and store glucose. And the liver will store this glucose in the form of glycogen. You may recall that glycogen is a polysaccharide. Glycogen actually means animal starch. Starch is a polysaccharide. A large fraction of the glucose that's absorbed from the small intestine is going to be immediately taken up by these hepatocytes. And the hepatocytes are going to convert it into the storage polymer glycogen. A polymer is a molecule that's made up of repeating subunits. Glycogen is a polymer. It's made up of glucose molecules that are linked together, so repeating glucose molecules. So remember, out of the intestines, the hepatic portal system is going to transport the blood immediately into the liver. It's going to have a high concentration of glucose. Insulin is going to stimulate the liver cells to take up that glucose, and the cells are going to immediately then convert the glucose once it enters the cells into glycogen and store it. The insulin has several effects on the liver, however, that stimulate this glycogen synthesis to occur. One of the things is that as soon as the glucose enters the liver cells, the hepatocytes, it will be immediately attached to a phosphate group. This is called phosphorylation. This phosphorylation locks the glucose inside liver cells, because you may recall the bidirectional transporters, glucose transporters, that can transport glucose both ways across the plasma membrane. Liver cells have these bidirectional transporters. So if the glucose comes in, the bidirectional transporters would transport it right back out again if it wasn't tagged with the phosphate group, and therefore not able to get out of the liver cells. In order for the bidirectional transporters to transport the glucose back out, that phosphate group will have to be removed. In addition, several enzymes will become activated, and these enzymes will catalyze the covalent bonds that are necessary to attach the glucose molecules together in the formation of glycogen. So the net effect is pretty clear then. When the supply of glucose is abundant, insulin, secreted by the pancreas, tells the liver, when it binds to the insulin receptors, to bank as much of that glucose as possible and store it for later use. For a rainy day, you might say. In the absence of insulin, glycogen synthesis in the liver is going to cease. So if there's no insulin on the receptors, this whole process of forming glycogen as glucose enters is going to cease. And the enzymes responsible for the breakdown of glycogen, will then become active. If there's not much insulin, then insulin binding doesn't occur to cells. That must mean the blood glucose level is a little bit lower. So what's going to happen is this is going to stimulate the liver to reverse the process. The enzymes that form glycogen are deactivated, while enzymes that cleave glycogen into individual glucose units are going to become activated. The individual glucose units, once formed, are going to be dephosphorylated, and then those individual glucose units can be transported back out of the liver cells and into the blood by the bidirectional transporters. In addition to insulin, the pancreas produces a second hormone called glucagon, and glucagon has an effect on carbohydrate metabolism as well. So let's take a look at glucagon and its effect on carbohydrate metabolism. So glycogen breakdown is stimulated not only by the absence of insulin, remember our discussion in the previous slide, but also by the presence of glucagon. Glucagon is secreted when blood glucose levels fall below the normal range, under that range of below 90 milligrams percent. During this time, glucagon will then, once it's produced, stimulate the breakdown of glycogen stored in the liver. So not only, we're saying, will the absence of insulin stimulate the breakdown of glycogen, but glucagon will also stimulate the breakdown of glycogen. It'll sort of enhance or increase the rate of breakdown of glycogen inside those hepatocytes, those liver cells. In addition to this, glucagon will also activate hepatic gluconeogenesis. Gluconeogenesis means neo, new, genesis to generate, and glucose, to generate new glucose. Oftentimes what the liver will do under the stimulation from glucagon is take other substances like amino acids and convert them into glucose, which it can continue to pump out into the blood then, so that the blood glucose levels can come back up if they don't come up adequately by simply breaking down glycogen and releasing glucose. So what controls glucagon secretion? Glucagon is actually secreted by cells in the pancreas called alpha cells. These are in the pancreatic islets. They secrete the glucagon in response to hypoglycemia, or low blood concentrations of glucose. Two other conditions, however, are also known to trigger glucagon secretion. One of them is elevated blood levels of amino acids, such as would be seen after the consumption of a protein-rich meal. So if you eat a meal that's high in protein, doesn't have much carbs in it, you may have to get more carbs back into your blood, and the high levels of amino acids will be transported into the liver. The liver will say, what do we do with these? Glucagon will say, well, we don't have as much glucose as we should, so let's convert some of these amino acids into glucose and then transport it back out into the blood. In addition, we know that exercise can also trigger glucagon secretion, though we're not real sure why. It may be that because exercise causes the muscle cells to take up a lot of glucose, blood glucose levels go down and therefore glucagon is released, but we just don't know for sure. Okay, so once the glucose enters the cells, how do the cells metabolize the glucose? Well, all cells metabolize glucose through a process called cellular respiration. Cellular respiration has three distinct phases. These phases are called glycolysis, the Krebs cycle, and the electron transport system, also known as oxidative phosphorylation. Now, it's not my desire here to teach the whole of cellular respiration. You probably learned about cellular respiration in a basic biology class or maybe even in A&P 1. So I'm going to use just this real simple diagram to kind of give you kind of an overview of what's going on here, and maybe some things will pop into your memory about how this occurs. So glycolysis, the first phase, as we can see here, is a phase in which once glucose molecules enter cells, during glycolysis in the cytoplasm, there are enzymes that will attack the glucose and break down the glucose into a 3-carbon compound called pyruvate. As a matter of fact, glucose is a 6-carbon compound that gets split into 2 3-carbon pyruvate molecules. During this process, glucose, as it's being broken down, releases hydrogens. These hydrogens are picked up and carried by a substance called NAD to form NADH. The hydrogens via NADH will be transported into this yellow structure here, represents the mitochondrion. These electrons, these hydrogen electrons and atoms, will be transported into the mitochondrion, and they'll enter what we call the electron transport chain. The pyruvates will now be able to pass through the outer membrane of the mitochondrion and enter the matrix inside the mitochondrion. Once inside the mitochondrion, these pyruvate molecules will be attacked by enzymes, and the pyruvate, representing the remainder of the glucose molecule, will be broken down or completely dismantled. By the way, out here during glycolysis, remember that when bonds are broken, energy is going to be released. When the bonds are broken that cause glucose to be converted into 2 pyruvate molecules, enough energy is released to form 4 molecules of ATP. But actually, we get a net of 2 molecules of ATP during this process because it requires 2 molecules of ATP to actually break the glucose down in the first place. So 2 molecules to break it down, 4 molecules released as a result. We get a net of 2 molecules of ATP. Back to now, once the pyruvate molecules enter the mitochondrion, they undergo a process, a series of chemical reactions referred to as the Krebs cycle, named for the scientist who first described it. During the Krebs cycle, as I said, the pyruvate molecules are completely dismantled, bonds are broken, energy is released, and as a result, the energy can be used to form more molecules of ATP. There's a net gain of 1 more molecule of ATP, we say, per turn of the Krebs cycle. That is, for each pyruvate molecule that enters. So for a single glucose, there are going to be 2 pyruvate molecules that enter the Krebs cycle, and each one of those, the dismantling of each one of those pyruvate molecules is going to lead to 1 ATP, or 2 molecules of ATP total. Not to forget that as the pyruvate molecules are dismantled, hydrogens are going to be released again. These hydrogens are now picked up by one of 2 carriers, either NAD, which we've seen out in glycolysis to form NADH, or FAD, a new carrier that's found here only in the mitochondrium to form FADH2. And again, like the NADH from glycolysis, these NADH and FADH2 molecules are going to transport hydrogens into what we call the electron transport chain, or oxidative phosphorylation. And it's a pretty complicated process, but to break it down, basically, these NADs and FADs deliver their hydrogens to this electron transport chain. The hydrogens are stripped of their electrons, and the electrons are used to manufacture more ATP via an enzyme called ATP synthase. Here, most of the ATP during cellular respiration is produced. There is 30 molecules of ATP that are going to be produced from the transport of the NADH, and 4 molecules of ATP that are going to be produced from the transport of the FADH2 molecules. So this is going to give us a net gain of 34, 35, 36, 37, actually about 36 molecules of ATP. Okay, if you need to understand more about this process of cellular respiration, here's a great website that you can go to that'll help you to understand more on how glucose is used inside cells to produce energy. Don't forget, this energy, this ATP that's produced then, is going to be used to power up the chemical reactions that occur inside the cells. Let's go on to the next slide. All right, so we said that insulin has profound effects on carbohydrate metabolism, also on lipid metabolism and protein metabolism. Let's take a look at lipid metabolism now. So insulin promotes the synthesis of fatty acids in the liver. Ah, that's interesting. So when the liver is saturated with glycogen, it's taken up as much glucose as it can, and it's made as much glycogen to store as it can, and it just can't store anymore. There's no more room to store any more glycogen. What happens? Any additional glucose that's taken up by the hepatocytes is going to be shunted into pathways that lead to the synthesis of fatty acids. In other words, the hepatocytes are going to take the additional glucose from the glycogen and convert that glucose into fatty acids. Those fatty acids then are going to be exported from the liver, and they're exported primarily as lipoproteins. We'll talk more about lipoproteins in just a bit. Insulin also inhibits the breakdown of fat in adipose tissue. So the idea is that when insulin is released, it binds to the insulin receptor. Now, we know that one of the things that the insulin causes is for adipose cells to take up excess glucose from the blood. But in addition, if there's all this glucose that's out in the blood, and glucose can be used to provide energy for cells, then there's no reason for adipose cells to release fat, because fat is also an energy compound. Whenever it's released into the blood, it can be taken up by cells and used to carry out cellular respiration. We'll see that in a little bit. So there's no sense in releasing fat into the blood when there's already a lot of glucose in the blood. So insulin's also going to have the effect of inhibiting the breakdown of fat in adipose tissue and its release into the blood. The insulin, it actually inhibits the intracellular enzyme that breaks down triglycerides to cause them to release fatty acids. So insulin facilitates the entry of glucose into adipocytes, and within those cells, glucose can be used then to synthesize glycerol. Glycerol is a 3-carbon compound, so the glucose once again can be broken down, but this time instead of forming pyruvate, in fat cells it's broken down into a 3-carbon compound called glycerol. You may recall from AMP1 that a glycerol, now with 3 fatty acids attached to it, forms a triglyceride or a neutral fat. It forms the fat that can be stored inside the fat cells. Okay, let's talk for a minute about lipoproteins. So these fatty acids that are released by the liver, remember the liver's got this excess glycogen now. It's taken in all the glucose it can and stored as much glucose as it possibly can, and now it can't store anymore. So what happens? The excess glucose is converted into fatty acids, and these fatty acids then are released by the liver back into the blood. And these fatty acids are then transported via the blood in one of 2 ways back to the fat cells. One way they're transported is bound to the albumins, remember one of the 3 major proteins that are present in the blood. Or they can be bundled into lipoproteins. So the fatty acids don't have a positive or negative charge associated with them, and so the water in the plasma can't transport the fatty acids. So what happens is the fatty acids have to be transported in the blood by other means. One way, they can be picked up by albumins and attached to albumins which have a negative charge associated with them. The albumins then can be attached to water molecules and kind of drag the fatty acids along with them to the fat cells. On the other hand, the fatty acids can be bundled into lipoproteins. Lipoproteins are tiny spherical bodies that are composed of triglycerides, or fats, protein, and cholesterol. Lipoproteins, by the way, are divided into 3 classes according to their density. The first class are what we call very low density lipoproteins. These lipoproteins are about 60% fat and about 15% cholesterol. And cholesterol, because they have cholesterol in them, that's where they get their name, cholesterol. Okay. The second class are what we call low density lipoproteins, or LDLs. These are about 50% cholesterol, and a high level of LDL, low density lipoprotein in the blood, indicates there is much cholesterol. You can see there are about 50% cholesterol in the blood, and that puts a person at a higher risk of coronary artery disease. The third class, main class of lipoproteins, are what we call high density lipoproteins, or HDLs. HDLs are only about 20% cholesterol, they're about 5% triglycerides, and they're about 50% protein. High-density lipoproteins are oftentimes referred to as the good cholesterol because they transport cholesterol to the liver for metabolism, and they represent triglycerides or fat and cholesterol actually being cleared from the blood. So this is why they're considered to be the good ones. They're high density lipoproteins means lots of fat and cholesterol clearance from the blood. Low density lipoproteins, by the way, are the lipoproteins that transport fatty acids and cholesterol to the fat cells. So they're an indicator of high fat and cholesterol levels in the blood, and that's why they're considered the bad cholesterol. So a high concentration of HDLs then is associated with a lower potential for heart disease. You're clearing fat instead of storing fat, and the potential for fatty plaques to form in blood vessels that can block them and cause heart disease is much lower when high density lipoproteins are higher. I think this is a little bit out of order, but here's a good podcast that I posted on Blackboard that you can listen to and maybe get excited enough about to read this book called Good Calories, Bad Calories by Gary Taubes. And it talks about what we call glycemic index and foods that can lead to a lowering in insulin production and how that can affect health. So take a look at that podcast. For now, though, we can take a look at what we call the cholesterol profile. This is important for healthcare providers to understand. So over on here on the left side, we have the total cholesterol level. And if the total cholesterol level, we like it to be less than 200 milligrams per deciliter. This is considered to be desirable. And when the cholesterol level is below 200 milligrams per deciliter of blood, that's considered a lower risk for developing coronary heart disease. A cholesterol level from 200 to 239 is considered to be borderline high, and 240 and above is considered to be high blood cholesterol. That high blood cholesterol in that level puts the individual at more than twice the risk of coronary heart disease than if their cholesterol level was below 200 milligrams. So you can see why healthcare providers want you to have a total cholesterol under 200 milligrams per deciliter. Now we can break it out here on this slide into the HDL cholesterol and LDL cholesterol levels. So for men, ideally we like them to have a 60 milligrams per deciliter or above cholesterol level. This would be considered a high HDL cholesterol. An HDL of 60 milligrams per deciliter or above is considered protective against heart disease. So we'd like the higher the better. By the way, a HDL level, below 40 for men and below 50 for women is considered low HDL and is a major risk factor for developing heart disease. So what are your risk factors? We can break out the LDL and HDL and take a look at that. Now, the LDL cholesterol level, we like it to be as low as possible because remember, LDLs indicate the fact that fats and cholesterol are being transported into the blood and therefore can cause fatty plaques and occlusion of blood vessels. So we like the LDL level to be less than 100 milligrams per deciliter. That would be considered to be optimal. 100 milligrams to 129 milligrams would be near or above optimal. That means not quite as good. 130 to 159, borderline high. 160 to 189, remember this is just the LDL, is considered high. And 190 and above is considered very high. Now remember, we want the total cholesterol level, anything over 200 puts us borderline high to high. So these are numbers that are well worth making sure you understand and know. Okay, so now the question becomes, once fats enter cells, how do the cells use the fat? Okay, well, once the fat enters the cell, remember a fat is referred to as a triglyceride because is a glycerol with 3 fatty acids attached to it. The triglyceride is going to be broken down by enzymes into an individual glycerol and into 3 fatty acids. During cellular respiration, once glycerols form, remember they're 3 carbon compounds, they're easily converted into the 3 carbon compound pyruvate. And just as we saw in the cellular respiration of glucose, the pyruvate then can enter the mitochondrium, undergo a kind of an intermediate step. It'll be broken down from a 3 carbon compound into a 2 carbon compound, which is referred to as an acetyl group. The acetyl group will enter the Krebs cycle, and as they say on Seinfeld, yada, yada, yada. The Krebs cycle takes place and oxidative phosphorylation, or the electron transport chain. Fatty acids, on the other hand, will enter directly into the mitochondrion, and once inside the mitochondrion, those fatty acids will be broken down into the 2 carbon acetyl groups, and those 2 carbon acetyl groups will enter the Krebs cycle, and yada, yada, yada. Okay, that ends our screencast for today, and we'll take a look at insulin and protein metabolism and the rest of our PowerPoint next time.